We're continuing on with Chapter 11, looking at how Mendel continued his studies and arrived eventually at his law of independent assortment. So we're going to start with one of the experiments that led to this law of independent assortment. With this particular study, Mendel was looking at two heritable traits in the pea plants at the same time. So he already recognized that the traits of like flower color, um, smoothness or round wrinkledness of the seeds, seed coat color, etc., that those are all heritable traits. And recall that when he crossed the dominant and the recessive individuals, that sometimes in the F1 generation, the recessive traits were not showing up. And so that led to some questions also about whether or not the dominant traits and the recessive traits would always be inherited together. Like would a plant with purple flowers, the dominant trait for flower color, always have the smooth seeds, the dominant trait for that? Or could there be a purple flower plant with wrinkled seeds and a white flower plant with smooth or wrinkled? Could you have different combinations of those traits? So to get at that, he crossed a true breeding plant with two traits. In this case, we'll look at the roundness and the color of the peas. So we have a round yellow pea plant that's true breeding or homozygous for both of those traits, crossed with the fully homozygous recessive individual for those traits. So in this case, it's the wrinkled green peas. So we're going to work through this Punnett square and look at the resulting outcome of that to determine whether what Mendel thought was true. Can those dominant traits only be inherited together, or can you get all those different combinations of dominant and recessive versions of those traits? So here's how you would set up the Punnett square for this example. Notice that each row and column header needs to have one of each of the alleles for the two traits. So we need an R and a Y for each column and row header. Now this one's simplified because there's only one allele for each trait that each parent can donate. The first parent with the round yellow peas can only give a dominant allele for round and a dominant allele for yellow. So they're listed across the top, R, capital Y. And on the side, we have lowercase r and lowercase y from the wrinkled green pea plant parent. In the box with the expected genotypes for the offspring, the only possibility is where they get one of each allele from each parent for both traits. So it would be big R, big Y, little y. Therefore, the genotypic results are 100% big R, big Y, little y. So they are heterozygous for both traits. And the phenotypic results are that they are all 100%, all the offspring would be round with yellow peas. The peas would be round and yellow, that is. Okay, so this doesn't really get at the answer to Mendel's question because still the only possible outcome was the round and yellow peas, and those are both the dominant traits still. So like his first set of experiments that led him to his other findings and the law of segregation, he ran another round of experiments here where he let the F1 generation offspring self-pollinate. So let's show that Punnett square and examine the results and see if we can arrive at what he found that led to the law of independent assortment. So here's the setup for this next problem. And this is a dihybrid cross. Remember that a monohybrid cross is when you have a cross between two parents who are heterozygous for one trait. A dihybrid cross is when you have a cross between two parents who are heterozygous for two traits. So having big R, big Y by big R, big Y is a dihybrid cross. Now I just set up the Punnett square rows and columns so that we could talk about that. As you can see, it's much larger than we've done before. 4x4 Punnett square with 16 possible outcomes. So how did I get these row and column headers given the genotypes of the parents? Well, I like to use the FOIL method. If you remember from math class, first, outer, inner, last, to come at these combinations of alleles. And notice that every row and column header needs to have one R and one Y, just like we saw in the previous example. So let's apply the FOIL method to the first genotype here. So the first would be the first of each letter type. So we need an R and a Y. So I'm going to have big R, big Y. So those are listed here first. Both down the, because they're the same here, so that's why we can do this. So I have it across the row, in the first row header, and the first column header as well. Big R, big Y. Let's continue that process. So now if you look at the outside ones, it's going to be the very first letter R and the very last letter Y. So big R, lowercase y would be the outer using the FOIL method. Next is inner. So it's the two that are most inside next to each other, little r, big Y. And lastly, the last R and the last Y in each genotype. Little r, little y for both. Okay, I hope that helps you, especially when we deal with these more complex Punnett squares. So now that we've set up the rows and columns, now it's time to solve the Punnett square. Go ahead and see if you can do it. Pause the video and apply what we learned in the first example. Each box should have a full genotype for each trait, 2 R's and 2 Y's. All right, so here is the fully completed Punnett square. Now, instead of you going through one by one and comparing exactly what you have with mine, the best way actually to check it is to make sure we get the same phenotypic results. We're not going to do the genotypic results. Let's just cross that out because it's just a little too complicated and it's not really necessary. And we want to make sure that we get the correct answer for the phenotypic results. So what I recommend is I always start at the top left and I read this kind of like a book from left to right, from top to bottom, and we account for every possible cell in the Punnett square and what that phenotypic result will be. So looking at this first box, big R, big R, big Y, what would be the phenotype of that outcome? Well, recall that the dominant traits were round and yellow. So we want to go through the entire Punnett square now and count up how many outcomes of the total 16 will have round and yellow seeds. So what I like to do is check them off as I go so I don't double count anything. I will use a different color pen for this. So here we go. So we have anytime you have a dominant allele for both traits is going to be round yellow. So far I'm at 4, 5, 6, 7, 8, 9. So that means we have 9 out of 16. We're not going to convert that to a percentage. In fact, I want to keep it out of the same denominator so we can simplify it to a ratio that will always apply to a dihybrid cross for the phenotypic ratio. So 9 out of 16 is the probability of the offspring having round and yellow seeds. Now, wherever we left off would be the one we didn't check off next. So that would be big R, little y, little y. Let's count up how many of those. And these would be the round green round and green seeds. There's going to be 3 round green, 3 out of 16 probability of the offspring having round green seeds. All right, if you're feeling comfortable with this process, go ahead on with the next. Wherever we left off without any check marks, we have little r, big Y, big Y. This would be a offspring that has wrinkled yellow seeds. So go ahead and count them. You should get 3 out of 16. Leaving us with 1 out of 16 chance of the offspring having wrinkled green seeds. Okay, so now that's going to simplify. Oops. Okay, I'm not sure what just happened, but I can't get all that back. So go back and pause and make sure you gather all that data. But it's going to simplify to a 9 to 3 to 3 to 1 ratio. This will always be the phenotypic ratio for a dihybrid cross. So we did get that because remember it was 9 out of 16 to 3 out of 16 to 3 out of 16 to 1 out of 16. So since the denominators are all the same, we can reduce that to a 9 to 3 to 3 to 1 ratio. So as long as you came to that, then we can assume that we both did it correctly. And this is ultimately what Mendel found. Here's a maybe a more easy on the eye version of that, with some colors and some visuals there and showing that 9 to 3 to 3 to 1 ratio. So this goes hand in hand with what we just did, even though there might be some slight differences to how they laid it out. Okay, so now let's come back to what Mendel's conclusions were and how he ultimately arrived at his law of independent assortment. So as we already know from the law of segregation, the alleles segregate or separate during the formation of the gametes. So this happened to each pair of alleles in the 2 separate traits that he was looking at, the yellow versus green peas and the smooth versus wrinkled peas. Each pair of those alleles separated independently during the formation of the gametes. Now, this only applies to genes on different non-homologous chromosomes because there are such traits that are linked and sometimes are inherited together, but this was not the case for Mendel's pea plant studies. So as a visual, let's go back to this meiosis diagram. We saw this before when we were talking about the law of segregation, but now I want to focus on the one on the right side, the law of independent assortment, which states that alleles of genes on non-homologous chromosomes assort independently during the gamete formation. So as you can see in the results, all the gametes are genetically different. And that's because of what's happening in metaphase 1, where, and you maybe recall when we talked about this, that one of the sources of genetic variation in the gametes is how the tetrads of homologous chromosomes line up during metaphase 1 specifically, and how that's random, how they line up. Like the fact that on the left, in this scenario, both of the maternal pairs of sister chromatids are on the left side, and they're going to end up in the gametes together, versus in the scenario on the right, it's a mixed match of maternal and paternal chromosomes, and so all of the offspring gametes have one maternal and one paternal. So that is the law of independent assortment, but it's rooted in what happens as the chromosomes line up at metaphase in the tetrads in metaphase 1. And it could also apply to metaphase 2, but really when you have crossing over. These don't show crossing over, but if there was crossing over, that would also come into play as a source of genetic variation.